Assalamualaikum and a very good day to Dr. Haryati. Today here we will be presenting our group presentation on the title of Structural Determination of Anti-Influenza Drugs in Combating Influenza Viral Infection. So before we proceed, here I introduce our team members, which is Lau Siu King, Miruna binti Arifin, Siti Nur Shikira binti Jamaluddin, and also me, Nur Ashikin binti Muhammad Rasul. So our topic focus more on influenza virus. So this is the structure of the influenza virus. So influenza a virus is the major cause of seasonal or pandemic flu worldwide. So the potential of pandemic influenza outbreak has attracted many scientists and researchers out there to find and create any possible anti-influenza drugs in preventing influenza viral infection. So currently we will discuss two types of protein inhibitor, which is neuraminidase inhibitor and fusion inhibitor to be specific cyclic P7 peptide as an anti-influenza drugs as strategy to overcome the viral infection.
Assalamualaikum warahmatullahi wabarakatuh and a very good morning. My name is Siti Nur Shakira binti Jamaluddin and I represent you about the structural determination of a neuraminidase and fusion inhibitor. Before I begin, I would like to give a short summary on the process of the structure-based drug design. Okay, so these process are often proceeded through multiple cycles before the optimized lead goes into the phase clinical trial. The first cycle includes the cloning, purification for choice of the drug target, and proceed with the structural determination of this target protein by using either X-ray crystallography, NMR, or homology modeling. By using computer algorithm, compound or fragment of the compound from the database are positioned into a selected region of the structure. These compounds are scored and ranked based on their steric and acidic interaction with the target site, and the best compound will be tested with the biochemical assay. So in the second cycle, okay, basically we are going to synthesize, okay, the promising lead from the first cycle together with the target drug. So we are going to combine them and we are going to look for their structure determination, and then we can analyze their interaction. Okay, after that, we are going to also do optimization in order to increase the potency. So additional cycle can be included by synthesize the optimized lead. We do the structure determination again, and we can carry out a further optimization. So actually, at the end, after several cycles of the drug design process, the optimized compound usually show a marked improvement in the binding and often specificity for the target. So I would like to proceed with next actually for the structural determination in neuraminidase by using X-ray crystallography. So the influenza neuraminidase was recognized, okay, as a potential drug target in the 1970s. The structure showed a large and highly conserved active site that appeared to be quite rigid, making it an ideal target for the structure-based drug design. Basically, neuraminidase, or we call as NA here, is a tetramer of a 4 identical polypeptide. Each polypeptide contains about 470 amino acids arranged in a 4 domain. From this crystallography, actually, we can identify that a positive charge consists of arginine 118, 292, 371, which interact with the carboxylate group of the inhibitor, and the negative charge consisting of the glue 119, glue 27, sp 151 interact with the guanidine or the amino group of the inhibitor. Moreover, okay, actually the presence of the 150 loop from the residue 147 until 152 actually have some adjacent to the active site, which actually can help in the binding of the inhibitor by formation of the closed and open conformation. Okay, so next I would like to continue with the NMR analysis by using one of the neuraminidase inhibitor, which is oseltamivir. So we are going to look for the interaction of this inhibitor with the neuraminidase, and we are going to actually also review maybe the mechanism, but we are going to use a model membrane prepared by using the MDC. Sorry, we are going to use the MPC. Actually, the process is easy. Okay, actually we are going to take a quantity of the NPC and then we dissolve them in the chloroform. After that, the solvent was evaporate with a stream of nitrogen so that we can deposit a lipid film of the wall of the container. Then the last trace of the solvent were removed by vacuum drying for at least 1 hour, and then the film was hydrated with the required amount of the solution GS1404. Okay, GS1404 is actually oseltamivir, okay, the neuraminidase inhibitor. Uh, and this we are going to hydrate them in the D2O, the hard water, and then we are going to incubate in the water bath at 50 degrees Celsius every repeating vortex. Okay, so we are going to use the concentration, the membrane, the lipid, we're going to use 100 millimolar, while the inhibitor we are going to use 10 millimolar. And then we are going to do this in the unilamellar vesicle, which we are going to sonicate the above dispersion with a Branson sonicator 450 at 50% duty cycle, and until we can get the optical clarity. After that, we are going to record all the NMR experiment by using Bruker Advanced at the 500 megahertz spectrometer. So we going to do the 2D noisy experiment. Okay, and we are going to carry out the NMR in a realization delay of 1 second with a broadband proton decoupling. So this is the result. Okay, um, the carbon, so you can see here, okay, actually there are about G spectrum here. Okay, we have the first one is actually only the membrane, okay, the NPC only, and then the second one is actually only the inhibited oseltamivir inside the hard water, and the last one we actually combine two, okay, the NPC all and also the inhibitor oseltamivir. So actually we can see here actually the carbon 33 spectrum for the unilamellar DMPC vesicle as well as the DS4104 in the D2O actually can be characterized by sharp lines, okay, and on the incorporation of the GS4104 into DMPC unilamellar vesicle, the lipid signal continue to remain sharp, while okay the GS4104 signal show differential broadening. This can be put into three categories, okay, signal which are remain sharp but are shifter, and then those are broad and shifter. Finally, those are broad beyond detection. Okay, so this differential broadening of the GS41 1404 sorry 4104 actually resonance arise because of the different part of the drug bind to the lipid bilayer with different affinity. So we can see the interaction here. So next we are going to do we have a 2D noisy spectrum of the GS4104 incorporate with the MPC unicellular vesicle. Okay, so such spectrum actually can provide us information on intermolecular interaction from the intermolecular noise between two molecules. So these noise actually arise when the two atoms with either within the molecule or from a two different molecule are at distance less than 5 angstrom. So basically, these GS4104 resonance are indicated in the figure along F2 dimension here, and the intermolecular noise between lipid and GS4104 are indicated in the F1 dimension here. Okay, and this is the result we can see here. So we can actually conclude from the observation, it can tell us that GS4104 is actually used as a hydrophobic chain, and also the cyclohexene group to bind via hydrophobic interaction with the lipid spanning both of the head group as well as the interior of the lipid bilayer. So actually we can conclude here oseltamivir actually can interrupt the membrane fusion. So what is membrane fusion? Okay, membrane fusion is actually a process of the in the biological system, and which involve with the union of two opposite bilayer in order to complete the process, such as like hydrocytosis or viral infection. And these oseltamivir actually have a interfere with the cell fusion. Okay, so it can also interfere with the virus entry step. So another drug, another drug that can be used, actually Abido, as a fusion inhibitor. So this is the NMR analysis. Okay, here we can see this black line actually only the abidol, in the hard water, and here is actually the mixture of the abidol and also the uh, model membrane, uh, which are prepared by using the MPC. So based on the result, actually we can see that abidol interaction with lipid induce chemical shift changes in the abidol resonance when compared to the one that observed in the water, which means there is some interaction happen that actually can inhibit this fusion. And this also make it Abido as potential drug for the antiviral. Okay, so that's all from me, and thank you.
So my name is Nur Ashikin. Next, I will further elaborate on the structure of the two types of protein that are used in existing on design of drugs for treatment of disease. So first protein is neuraminidase. So neuraminidase is a type of protein, or also called as Na, that is a tetramer of four identical polypeptides that exist on variant surface. And the four monomers fall into four distinct structural domains that consist of head, stalk, transmembrane, and also cytoplasmic tail. So this is the structure of the neuraminidase. So we go through first to the structure of the Na itself. So the catalytic head of NAs consists of a box-shaped structure with four monomers, and each monomer is in the form of a six-bladed propeller structure, with each blade having four antiparallel beta sheets that are stabilized by disulfide bond and connected by loops. So a functional catalytic site is present on the surface of each monomer, and it arranged sideward rather than upward. This is to promote the ability to cleave the sialic acids from nearby membrane glycoprotein to prevent the virus trapping. So the active site is located in a deep central cavity in each of the four monomers that consist the active form of the neuraminidase. The active site is so important for virus spreading and is highly conserved, making it desirable target for neuraminidase inhibitors that can be used in antiviral therapies. So next is the transmembrane domain, which the N-terminal hydrophobic transmembrane domain attaches the Na to the viral envelope. So this attachment provides the signal for translocation from the endoplasmic reticulum to the apical surface, as well as the association with the lipid raft. So actually, both the cytoplasmic tail domain and the transmembrane domain contribute to the transport of the glycoprotein to the apical plasma membrane. So further with the stalk domain, so the cysteine residues at the neuraminidase stalk may assist with the tetramer stabilization by enabling disulfide bond to form between each monomeric Na unit. And the presence of the carbohydrate side chain within the stalk is thought to contribute further to the stability of the tetramer. So basically, the length of the stalk region can have a significant impact on particular virus characteristics. So previous study has proved that reduced stalk length will impact the Na activity of the variants because the reduced height may hinder access to cellular sialic acid expressing receptors, and that the towering HA blocks the shorter Na catalytic domain from gaining access to the sialic acid. So the lack of the Na function would trap the viral particle and prevent its attachment to the cell surface receptor for viral entry. So reduced stalk size may also alter the geometry and dynamics of the enzymatic pocket itself, modifying its affinity for sialic acid as shown in this figure on the next slide. So this figure shows the part A, the optimal Na stalk length enable the Na catalytic head access to the sialic acid expressing receptor, while for the figure in B shows that the reduced stalk length may impact the ability of the Na to contact sialic acid on the cellular receptor, as the neighboring HA may sterically hinder the shorter neuraminidase catalytic domain from gaining access to the sialic acid. So done with the neuraminidase protein. So we next, I continue with the second type of protein, which is cyclic peptides, or to be specific, we have discussed on cyclic P7 peptide in the next few slides. So this is the figure that shows the structure of the cyclic P7 peptide. So generally, the cyclic peptides are usually composed of 5 to 14 amino acids and have a molecular weight of about 500 to 2000 Dalton. So the moderate size and diverse functional groups of peptides ensure that the contact area is large enough to provide high selectivity and their potential to form multiple hydrogen bonds that can lead to strong binding affinity. So in addition, the cyclization of the peptides also facilitate the passage through the cell membrane, thus broadening the potential use of cyclic peptides in generating the structure and also the function features that can be used as the pharmaceutical agent. So because of that favorable characteristic features of that cyclic peptide, various cyclic peptides have been exploited for biomedical and other purposes that we'll be discussing more in the next few slides.
I am Lau Xiuqing, and now I will proceed the presentation with the importance of the reported structure in the design of drugs for treatment of influenza A. This figure shows influenza A virus. And my part of presentation will be focusing on neuraminidase. For your information, neuraminidase is one of the two major surface glycoproteins found on the influenza virus particles. Its function is to cleave off the terminal sialic acid from the cell surface glycoconjugates, which is the receptor for the other surface protein, hemagglutinin. It is known that inhibition of neuraminidase can effectively delay the release of progeny virions from the surface of infected cells. Therefore, there is a drug development targeting the neuraminidase protein, as it is proven to be a useful target for antiviral therapy because its active site is highly conserved in both amino acid sequence and spatial orientation, as well as it is prominently located on the surface of virion, so it will be easier to be targeted by the neuraminidase inhibitors. So this figure shows the interaction of the neuraminidase active site with sialic acid in the absence of neuraminidase inhibitors. Active site can be divided into 5 subsites, namely from S1 to S5. So S1, which is made up of arginine 118292371, will be able to interact with the carboxylate side chain of sialic acid. O4 hydroxyl can form interaction with residues glutamic acid 119, aspartic acid 151, and glutamic acid 227. Oxygen and nitrogen atoms of the acetamidol form hydrogen bonds with arginine 152, whereas the methyl group lies in a hydrophobic pocket near isoleucine 222 and tryptophan 178. O8 and O9 hydroxyl groups of the glycerol side chain are hydrogen bonded to the residue glutamic acid 276 and 277 to make an important contribution to the overall binding. So by studying well the interaction of the neuraminidase active site to the sialic acid, so there is a development of the neuraminidase inhibitors to act as a competitive inhibitor to stop the neuraminidase activity from interacting with the sialic acid. So there are 4 main examples of neuraminidase inhibitors lead to the drug development. So namely zenamivir, oseltamivir, peramivir, and laninamivir. So for zenamivir, oseltamivir, and peramivir, they are the derivative of the earliest transition state analog of the neuraminidase substrate 2,3-hydro-2-deoxy N-acetyl-neuraminic acid DANA, whereas laninamivir was produced via a modification of zenamivir. All of them mimic the substrate of neuraminidase, which is the sialic acid, and they can bind to the neuraminidase with higher affinity as compared to the sialic acid. So as we can see from this figure, so the development of these 4 types of inhibitors are actually based on the sialic acid because they have the basic structure similar to the sialic acid. And then those that are highlighted in red are the side chains or functional groups which is different from the sialic acid. And those side chains which makes the inhibitors to be able to have more interaction and higher binding affinity to the neuraminidase. So for zenamivir, it has a positively charged guanidinol group replacing the hydroxyl group that is linked to C4. So it increased the binding to the neuraminidase approximately 10,000-fold as compared to sialic acid. For oseltamivir, C4OH and glycerol side chain of DANA are replaced by amino group and pentyl ethyl ester group respectively. For peramivir, it is a cyclopentane derivative with a guanidinol group and pentyl ethyl ester group. For your information, cyclopentane is known to exhibit potent and selective inhibitory effects, especially on influenza virus neuraminidase. And for laninamivir, it is actually the modification of zenamivir where methylation occurs on the C7 hydroxyl group of zenamivir. So neuraminidase inhibitor structures are developed based on DANA or designed to mimic its binding to the neuraminidase, but differing structures results in interactions with different amino acid. So all of these inhibitors with the negatively charged carboxylate group are able to form strong hydrogen bonds interaction with the subsite 1 that consists of arginine 118,292,371. Guanidinol group, which is only present in the neuraminidase inhibitors like zenamivir, peramivir, and laninamivir, they can form stable hydrogen bonds and strong electrostatic interactions with the acidic groups of glutamic acid 119, aspartic acid 151, and glutamic acid 227. Glycerol side chain are hydrogen bonded to the residue, glutamic acid 276 and 277, to make an important contribution to the overall binding of neuraminidase to the inhibitors. Oxygen and nitrogen atoms of the acetamidol can form hydrogen bonds with arginine 152, whereas the methyl groups lies in a hydrophobic pocket near isoleucine 222 and tryptophan 178. So as shown in this figure, we can see that neuraminidase inhibitors have more interactions and higher binding affinity to the neuraminidase as compared to the sialic acid. Therefore, the presence of neuraminidase inhibitors will act as competitive inhibitors to the neuraminidase. Neuraminidase inhibitors, which mimic sialic acid structure in its transition state during Na-catalyzed hydrolysis, represent a class of effective anti-influenza drugs. So as I just explained, they are zenamivir, oseltamivir, peramivir, and laninamivir. So for zenamivir, it is mainly delivered through inhalation and was licensed worldwide since 1999. For oseltamivir, it is mainly administered through oral and was licensed worldwide since 1999. For peramivir, it is an intravenous formulation that is developed only for compassionate use and was licensed in few countries like Japan, South Korea, China, and USA since 2014. For laninamivir, which is the modification of zenamivir, was only licensed in Japan since 2010 and is currently in phase 3 clinical trials in USA. As a summary, after budding, virions need to detach from the host cell by viral neuraminidase, which possess the sialidase activity, to cleave off the sialic acid, thus releasing the virions progenies. So this step is the target of the anti-influenza neuraminidase inhibitors that are used in clinical practice. So in the presence of neuraminidase inhibitors, it can stop the release of viruses from the infected host cells and prevents the new host cells from being infected. Thus, it can suppress the viral population and allow the time for the host immune system to eliminate the virus and eventually control the infection.
Good day to Dr. Haryati. So today I will be presenting on the importance of fusion inhibitors and the ways they could be incorporated when designing drugs which can actually combat the influenza A virus, which has been a very upcoming conundrum across the globe, which really requires a very insistent and eminent sort of treatment to this, which the research community is currently working on. So before I present, I would like to give some key points for whatever that I'm going to present today, a light brief highlights. So first of all, we will look into the viral mechanism, the particular machinery, which actually how it really propagates throughout the cell and how it replicates and so on, and which machinery has been targeted by the scientist community on the whole by using infusion inhibitors, and how do they incorporate them into drugs which sometimes have the potential to become successful drugs, and also the drugs which have been successfully established amidst the market. So first and foremost, as I said, the viral hemagglutinin machinery as an antiviral target. So this particular machinery has been targeted as an antiviral mechanism by the research community themselves to provide medicines, provide drugs to combat the influenza A virus on the whole. So let me brief. First of all, these viral hemagglutinin proteins, these particular proteins, they tend to have this particular domain that bind to the glycans that are found in the cell surface of the sialic acid, which literally happens over here. So when they bind, what happens is internalization of the virions via endocytosis will lead to the decrease of the pH, which means the entire environment tends to be acidic amidst the endosome. So this is the turning point, or this can be marked as the climax, which actually leads to the subsequent effect or the tailing effect, the repercussions, which is caused by the virions, which actually tends to lead to the final replication, because this particular acidic environment could lead to the irreversible conformational change of the HA protein. So when there is a conformational change, the fusion protein, the fusion peptide of this particular HA protein is exposed. When it is exposed, this triggers the fusion with the endosomal membrane of host cell, and thus the viral ribonuclear proteins are released into the cytosol. They migrate to the nucleus for subsequent replication, and the entire cell is done by the virus itself. So scientists have targeted two important factors here. One is blocking of the HA binding to the sialic acid residue, which is the beginning or the initiation of the entire process itself. Or another target could be the blocking of the HA conformation change, which roots out from the acidic environment of the endosome itself. Hence, this is particularly the rough idea of what really happens due to the HA mechanism of the virus itself. So the internalization of virion could increase the acidity in the endosome, which will lead to the conformational change, and thus the repercussions would be the membrane fusion followed by replications. So what do scientists do literally, and what do they have discovered is that this particular fusion inhibitors, how do they function? Now they function by preventing this acid-triggered conformational change, which then enables them to incorporate these fusion inhibitors when designing drugs as the antiviral drugs to combat influenza A virus. So the first fusion inhibitor, which I'd like to talk about, is P7 cyclic peptide, which is currently on the stage or the status of being a potential fusion inhibitor to be incorporated into drugs to combat influenza A virus. So from a very pivotal anecdote, P7 cyclic peptide is well prominent, well eminent for its specificity. Why? Because the structure of this peptide is solely conjectured on the note of the heavy chain complementarity determining region loop of BNEB F6V3 and framework region 3 of CR9114. So these components in this particular cyclic peptide makes it more specific, which makes it to be highly eminent and a highly potential candidate to be incorporated for drugs that are actually against influenza A virus. So looking in depth, note on a very in-depth sort of deliberation. P7 literally binds to the HA stem epitope. So this is where it hits the key point, the climax, where it blocks the low pH, the acidic pH, which is actually the prime factor which causes the conformational arrangements, followed by the membrane fusion, followed by replications. So this is particularly the first step in which the P7 cyclic peptide attacks or combats. It combats the low pH induced conformational rearrangements first. It blocks that entire process. by its specificity, by binding to the highly conserved HA syn epitope. So what makes it P7, what makes P7 super specific for group 1 HAs? It's because of the glycosylation of ASN38 in group 2 HAs and substitution of tyrosine 49 group 1 for a larger ASN group 2 that improves binding. These groups are literally which belongs to the HA itself, 2 different types of HAs, okay? So this particular figure here, it shows the structural elucidation of the group 1 HA specificity of the peptides. Moving forward, Arbidol. Now, Arbidol is one of the most successful fusion inhibitor, which has been incorporated into drugs successfully amidst the market to prevent influenza virus, to combat influenza virus, and it's one of the most successful feats by the scientist community themselves. So, applauses to them. Well, when we come to Arbidol, this particular topic, literally we will be focusing more on the hydrophobic cavity found in the HA trimer. This hydrophobic cavity in the HA trimer would be literally targeted by this particular Arbidol. So Arbidol works by actually targeting the hydrophobic cavity in the HA trimer, which is the particular place where it binds. So this particular binding literally increases its specificity on greater levels, on greater heights. So where does it bind? Let's look into a closer note. Arbidol consists of peptides that function as fusion inhibitors such as F1, F5, F7, and F8. So all these components all these peptides, they tend to bind in the hydrophobic cavity in the HA trimer stem at the interface between 2 protomers. And binding of this Arbidol to the cavity, hydrophobic cavity in the HA trimer will literally it will make some hydrophobic interactions for sure, and it will induce some conformational rearrangements to form a network of inter- and intraprotomal salt bridges, which will then subsequently or concurrently, it would literally block the pH-induced conformational rearrangements. So it will block the entire network by causing a different sort of conformational rearrangement, which is the formation of this particular intraprotomal salt bridges. So by that, it stabilizes the perfusion conformation of HA that inhibits the large conformational rearrangements, which will lead to the membrane fusion. That's the functions of, those are the functions of Arbidol. So Arbidol has a very high shape complementarity with the binding sites in these 2 groups of HAs, group 1, group 2. It has a very high complementarity due to the presence of F7 and F8 that work as a minor fusion inhibitors. So all these structural informations, all these opulent informations on Arbidol has really enhanced or enabled the scientist community to come up with superfluous drugs that can combat influenza A virus on a very high note. So incorporation of these sort of fusion inhibitors are really imminent and really significant, and the scientist community is still conforming with its efforts going forward and progressing with its efforts by literally researching more in depth on the structures of Arbidol, P7 cyclic peptide, and many sorts of other fusion inhibitors to be incorporated into the drugs and to come out with really efficacious drugs to put an entire full stop to the influenza A virus, though it might sound a little, well, perhaps not necessarily impossible, but let's just hope for the best and let's save our globe from this particular influenza A virus. Thank you.